Okay. So, biotechnology and its application. Now, in the previous chapter, we have already learned about the term and what are the equipments or what are the processes which we use in biotechnology for certain applications. Okay. So, in this chapter, we'll be dealing with the three categories over here. Okay. That is the agriculture, and then we'll be dealing with the medicine, as well as we'll be dealing with what the transgenic animals. Plus, we'll be seeing what are the ethical issues which we face when we use the biotechnology. Okay, so usually this is a particular stream which uses what biology as well as technology together. So because of that, whatever the outcome it gives you is more as compared to giving what individually, whatever is given by biology as well as the technology. So this thing is usually used in a industrial scale to obtain what a major production. For example, we can see that in the biopharmaceutical companies, therapeutics, diagnostics, okay, to process whatever the foods are there in the industry to make it more, to have more shelf life, okay, for that particular food, whatever is there. For all these purposes, we can use the biotechnology. So usually, if we see over here, they have what? Three critical research areas. Now, already I told you the three applications which were there. These three areas mainly use this technology. Okay. So now what is the main reason why they use the biotechnology in these categories? So the first thing is what? Providing the best catalyst in the form of improved organism, usually a microbe or enzyme. Now, already we know enzyme is what? Almost a catalyst. So it increases the rate of reaction. So whatever the process is there, it speeds up and you get the product easily, right? So this is the first thing for which we use the biotechnology. Second is what? Creating optimal conditions through engineering for a catalyst to act. So not only we are using a catalyst in the form of a microbe or an enzyme, but also we use certain engineering techniques, okay, which together what they will do is give us a greater outcome, whatever is there. Third one is the downstream processing technologies. Okay, now usually whatever the products have been made, whether you take the pharmaceutical companies or the food industries, right? Everywhere over here, the process, there is a long process which undergoes to get a particular medicine or whatever things are there. So those things are much more what? Rectified, as well as you get more purified product when you use a biotechnology in over there. Okay, so these are the three areas which we will be dealing over here. So the first one which is there is the agricultural field. Okay, now usually the farmers, okay, when they grow crops, they use the fertilizers, the pesticides, okay, many things they use, but they are expensive comparatively. But in turn, what happens is even if they are using the pesticides, insecticides, or whatever is there to kill the insects, okay, or to keep the weeds from growing back again, still what happens is there are almost more than 80% that it will come back again. So here biotechnology plays a role. Okay. And even though it's a one-time investment, but further whatever products they are going to get, it will give them what? More outcome comparatively to the basic resource that they were using. Okay, like the pesticides or whatever is there. And plus they also cause what? Environmental pollution. So that is also taken care of over here. Next one is what? The organic culture. Now, organic culture, we all know it's very expensive, right? So then to avoid this expense, what we will do is we will combine it with the genetically engineered crops. So in markets, we can see GM cotton, okay, or hybrid tomatoes, hybrid brinjals, all these things are there. So if you go to a market, you will see many categories. There will be locally grown, there will be organic foods, there will be also what? Genetically modified foods. So genetically engineered or modified plants are what we are going to see in this chapter. So this things deals with what? Generally the plants, bacteria, fungi, and animals. So genetic modification means what? The genes are manipulated over here, or they are what? Altered so that whatever the products which we get will be in a more purified or higher version forms we will get over there. So what is the advantage of using this particular GM plants? Okay. So if you see over here, the first one is what? It makes crops more tolerant to abiotic stress. So abiotic stress means what? It will include the droughts, heat, cold, all the environmental conditions which are there, which may make that particular crop not grow properly. So there this comes into outcome. Next is what? Pest resistant crops reduce the use of chemical pesticides. See, now generally in GM plants, what we do is you use a what do you say, organism. Okay, and from there you isolate a particular cell, and that cell will act in to prevent the plants from getting infestated. Okay, that process we are going to study further in this chapter. So pest resistant crops also you're going to get, and there will be a lesser use of the chemical pesticides as well. Next is what? It reduces the post-harvest losses. Okay, see, now obviously, compared to the post-harvest losses will be more, because if these abiotic stresses come into existence, then everything will be what? Into more loss. So that also can be prevented over here. Fourth one is it increases the efficiency of mineral usage by the plants. So generally, there are some plants which couldn't take the minerals or absorb the minerals via their roots from the soil easily. So if you alter the genes of that roots, okay, then basically what is going to happen is the absorption of the minerals also will be increased, and it prevents what? The early exhaustion of the soil fertility. Because as you know, annual crops, if you're doing, or perennial crop, whatever things are there. So when you do it yearly or monthly basis, okay, whatever land you're going to use again and again, it will be exhausted with the minerals. So by using a genetically modified crop, what will happen is the efficiency of its usage will also get increased, and the soil fertility also will remain unexhausted for a longer period of time. Then you have what? It enhances the nutritional value of the food. And the best example which is available is what? The golden rice. Okay, that is fortified with what? Vitamin A. Now, generally in the shops when you go, okay, or to purchase the groceries or vegetable shop, whatever, wherever you go, there will be what? A higher version of every food. So those are nothing but they will be fortified externally with what? The vitamins or minerals, whatever is there, which is not initially present in that particular plant. So you can also increase that capacity over there by using the genetic modification. And the last one is what? To create tailor-made plants to supply alternative resources. Now, it is not necessary that from one plant you're going to get just one product. Okay, so there will be byproducts also which you will be getting, right? So whatever the alternative resources which is written over here, like starch, fuel, pharmaceuticals, etc., you will get what? A much superior supply to all those particular resources by using this technique. So these are mostly the advantages of what? Genetic modifications, what when you do it in the plants. Okay, so first thing which you have is what? The pest resistant plants. Now, pest, okay, which is there, it's a very big concern for the farmers as well. So, when you use a genetic modification, that is when you alter the plant gene, okay, at that time, what will happen is that gene itself will act as a biopesticide. So, whatever the need for your insecticides or pesticides are there, that is also going to reduce. So nowadays, whatever the pest-resistant plants are there, they are in the form of what? BT cotton, corn, rice, tomato, potato, soybean, and many more are there over there. Okay, so first let's see about what? The BT cotton. Now, in order to know how actually there is an alteration of the gene, you should know that what is BT, okay, and what happens to the insects or the pests after inserting this particular BT gene into the plant. Okay, so BT means nothing but it's the short form for the organism Bacillus thuringiensis, okay, which you can see over here, Bacillus thuringiensis. So now usually whatever the genes you're going to take over here, they all will be what? Insect group specific. Okay, so hence whatever the strains of this BT, that is the Bacillus thuringiensis are there, they produce a protein. And that protein is initially responsible to kill your any type of worms or insects which is going to attack your plant. Okay, so this form of protein is called as what? Insecticidal protein, which is also termed as Bt toxin. Okay, so this particular insecticidal protein or the Bt toxin, which is there, it's a crystal form which you get. Okay, but initially it will be in an inactive form. Only once it enters inside the stomach of the insect or a pest, then only it gets activated. How? That we are going to see now. Okay, so how this phenomenon works of this Bt gene, okay, that is Bacillus thuringiensis gene is, usually what happens is this Bacillus thuringiensis genes, they have what? Certain kinds of or strains of the proteins. And that proteins, they are called as what? Insecticidal proteins or the Bt toxin, which are in the crystals form. So usually whenever they become mature, at that time, what we do is in their inactive form only, we will extract them. So that is called as what? Inactive protoxins. Okay. So whenever a beetle or any other pest comes to eat the leaves or any other parts of the plant, what is going to happen is they will take in that particular toxin. Means they are going to eat or ingest that particular insecticidal protein. Okay, so when they will ingest that particular insecticidal protein, what will happen is it will enter into their gut. Okay, and in their gut, they will be having what? An alkaline pH. So in the alkaline pH, what happens to the beta toxin crystals are they will solubilize themselves and they will get activated. So once they get activated, whatever the toxins which they have been ingested, okay, they will go attach to the surface of the mid-gut epithelial cells. They will create holes in the intestine of that particular pest or whatever the insects are there. And it will cause swelling of the cell and ultimately it will lead to what? Death or the lysis of that particular insect. So here if you see, this is how it takes place. You can see over here, it's a Bt spore or a crystalline toxin. So this particular caterpillar, okay, it's eating that particular leaf. So the normal gut bacteria, along with that, the crystal proteins have been taken in, and afterwards you can see at last what happened to that caterpillar? Lysis. Okay, because spores were formed, so it led ultimately to the death of that particular caterpillar. Okay, so usually they get infectious, and that is how they die. Same happens with whatever, how many numbers of the insects are invading the plant. Okay, the same mechanism happens with every organism, whatever is there. So this is how what Bt cotton was ultimately protected from the caterpillars. So usually whatever the toxin is there, it is coded by a gene which is called as what? Cry gene, CRY gene. Okay, so this is the toxin code which is there. So there are actually many number of genes which are acting against the insects. So according to that, we have the categories like Cry1AB. Okay, here you can see Cry1AB, Cry1AC. Okay, like that, many categories you will be having. So each category kills a particular type of worm. For example, if you take the Cry1AC or AB, it will control what? The cotton ball worms. Okay, if you take the Cry1AB, it will control the corn borer, the corn which are there, right? Those insects which attack on the corns, they will kill those particular insects. Okay, so according to what type of insect they are going to attack on, these codes are based upon that. So general code is the cry genes. They are responsible for what? Whatever the toxin production, that is the Bt, Bacillus thuringiensis. So this Bt cotton was the first introduced transgenic crop, okay, and later on, rest everything came. Like the second one, which you're going to see over here, is about the tobacco. Okay, so in the tobacco, usually we have a nematode resistance. We need it in the tobacco because nematode is nothing but in the roots of the plants. If you see, okay, there will be small nodules. Now, some nodules, they are very useful. Like if it's a rhizobium or something, it is very useful in the nitrogen fixation or other purposes. But when the nematode is in large number, what happens is ultimately that plant dies. Okay, it gets infected with the viruses and you won't get anything in return. So to make it resistant from the nematodes, okay, there was a technology used by the biotechnology, which is called as RNA interface. That is RNAi. Okay, so the first transgenic plant, which was the tobacco, it contained what? A resistant gene which was against the VD side. Okay, and that VD side name was what? Glyphosate. Okay, so to do this, they use the process which is called as RNA interface. So RNA interface is nothing but here there is generally the slicing of the mRNA. So specific mRNA which is responsible for the formation of the nematodes, right? So those are being silenced over there, okay? Or the slicing of that particular mRNA is being done, and in turn a complementary dsRNA is being produced. It's like a clone, okay? So complementary DNA, when it will get produced, it will bind to whatever the nematode RNA is there, and it will prevent it from the RNA, what do you say, replication. And further, what will happen is the nematodes won't be formed. So that is how actually the process works. So here, if you see the DNA, okay, it's converting to the mRNA and finally to the proteins. That is transcription and the translation process, right? So DNA to conversion of the mRNA includes a process which is called as transcription. And when the mRNA, that is the messenger RNA, converts into the protein, it happens through the translation process. Okay, so what happens is whatever the source of this. Yeah. So what I was saying is, yeah, this is the process how DNA to protein conversion actually takes place. Okay. So whatever the source of this complementary RNA is there may be chances that they are from an infected virus, or they are having the genomes of that RNA genomes of what? The infected virus. Okay. So what happens is we use this RNA interface, or we use this RNA as an intermediate to attack that particular complementary RNA, which is producing what? Infections. So generally to do this, you know that there are different kinds of vectors or plasmids are being used over here, right? So in this also, what we use is the agrobacterium, what vectors? Okay, so this agrobacterium vectors, and by using the nematode specific genes, okay, you will introduce this two into what? Your host plant, which is being attacked by the nematode. And then what is going to happen? It is going to produce the sense as well as antisense. Okay, whatever sense and antisense RNA is there, that will be produced in your host cells. And when you get the two complementary RNAs, and they will form what? From the RNA, they are going to form what? Again, the mRNA and then the end of the nematode over there. Okay, so this is how the infection is being eliminated by incorporating a vector with your nematode specific genes, which is going to ultimately kill that particular nematodes. Okay, so this is the second example. Apart from the BT cotton, protection from the caterpillar, the second one was what? Nematode resistance in tobacco plants. And this is how it happens. Okay, so this is all the two applications which we use in the agricultural field. Next you have is what? The application in medicine. Now this is a very wide application over here because pharmaceutical companies, they mostly use the biotechnology source to generate what? More powerful medicines by using specific genes or by altering what? The specific genes. So the most important technology which we use in this field is the recombinant DNA technology. Okay, so whatever the recombinant DNA technologies are there, they are having, what do you say, very immense impact in the area of your healthcare sector. So because of that, whatever the mass production is there, mass production of genetically engineered medicine, out of which the first one which comes is the insulin. Okay, so whatever the insulin is there, that was the first genetically engineered medicine, you can say. And that was created by using different techniques, and that techniques we will be seeing over here. So if you see over here, at present, whatever the 30 recombinant therapeutics have been approved, whereas out of 30, 12 are currently being marketed in India, means it's been used for various treatments, you can say. So let's see one by one. The first one which is there is the genetically engineered insulin. Okay. So now when you come to the genetically engineered insulin, now insulin, that is the organ which produces it, is nothing but the pancreas, right? So it is usually used for whom? Diabetic patients. So earlier, okay, before introduction to this particular genetics topic, in this insulin, which was used for diabetes, was extracted from where? This organ, which is the pancreas of the animals. And generally, the animals which they used were cattle and pigs. But what happened was when they used this extracted insulin and gave it to the diabetic human patients, it caused what? Allergy and certain reactions. Because as you know, in our body, there are two things: antigen, antibodies, right? So, whenever a recurrent antigen comes into your body, the body already has the antibodies present inside, right? So, it can detect and it will cure the thing. But if any foreign particle, a newly particle is being entered into your body, obviously your cells, they will be like calculating what kind of thing has been entered, something has come inside. We have to protect the cells or this human from that particular foreign thing. So uh, initially what does this do is it will cause what? Allergies, right? So in order to reduce this thing, what they did was they used to means bacteria to produce the insulin. Okay, so now you cannot directly isolate the insulin from the pancreas of the humans to avoid the allergies, right? So what is the other option which you have is by using the bacteria. Okay, so you can easily produce the human insulin by using the bacteria. Now, if you see the structure of the bacteria, okay, they usually contain what? Two short polypeptide chain. That is chain A and chain B. And these two chains are linked together by the disulfide bridges. Okay, so if you take the mammals, whatever insulin has been produced, before the insulin has been synthesized, they will be in what? A form called as pro-insulin form. That is, it's not activated yet. So it's a pro-hormone. So what happens is whenever it gets matured, then only the function of the insulin will be played, right? So if you see over here, in 1983, okay, an American company, they prepared what? Two DNA sequences which were corresponding to your initial whatever insulin structure, which was composed of what? Two chains, that is A and B. What they did, they introduced them into a vector that is a plasmid of E. coli, and they produced insulin chains over there. So the same structure which you initially have in your human insulin, that is A and B chain, which were binded together by the disulfide bonds, they created that in the laboratories. So how did they do this? This is how you have a proinsulin. They extracted it, kept it into a vector, which is a plasmid, which they took over here, went and what it did was whatever the diabetic patients were there, it was given like nowadays how we have the vaccine and all those things, right? So those ways only it has been incorporated into the human body and then it produces what? Insulin. So this is called as what? Genetically engineered insulin. For the diabetic patients, it could be used. Okay. Then the second thing which you have is the gene therapy. So what is this gene therapy? Okay, so usually gene therapy is nothing but what? It's a very good technique to correct a genetic defect. But when the child is in embryo form, it works better. Like for example, in a very advanced stage of medicine, if you use this gene therapy, you could even cure a baby which is already in the womb of the mother and it's having defect. Obviously, it's a very costly procedure and it's like not everywhere applicable also. Okay, still many things have to be done to give a better result of that particular defect. Okay, to do this, there was what? A gene therapy. So gene therapy is nothing but it's just a collection of methods which is just going to allow you to correct what? A faulty gene. So by using a healthy gene, you can correct a faulty gene and correct the function of that particular faulty gene, right? So how it's been done is whatever your genetic defect is there, that has been corrected by delivery of a normal gene to the. Now, for example, if you have a cell, okay, now in this cell, one cell is defective. So what you will do, you will take a normal cell, okay, which actually will replace this particular defective or faulty gene, and then it will take over the old gene and it will make it non-functional, and whatever the new gene you have incorporated via the gene therapy, it is going to function and it's going to repair that particular gene entirely, and it's going to give what a permanent cure for that particular defect. So one such trial was done actually, okay, in the year 1990, and it was done on a 4-year-old girl who was having what, adenosine deaminase deficiency, that is the ADA deficiency. So how this deficiency is caused is whenever there is whenever there is a deletion of a gene of this ADA, okay, at that time it will affect your immune system functioning. So one such cure, what you can do for this is by transplanting the bone marrow, okay, as well as by the enzyme replacement therapy, which is nothing but ADA injections you can give. But even though if you do any of the two methods, what will happen is it's not a complete cure for that particular girl. So for that, there came this gene therapy, okay, which completely or permanently cured this ADA deficiency in that particular girl. So what was done over here is they collected the lymphocytes from the patient's blood and they grew it in a culture in an aseptic condition. They just grew them, okay, and they introduced what, a functional ADA instead of that faulty ADA, and now they returned it to the patient and periodic checks were done, and they saw that whatever the ADA gene which was introduced from the marrow cells, okay, at the embryonic stages means what, initial starting of that particular defect, no, at that time when you inserted this particular ADA gene by using the gene therapy, it could give what, a permanent cure. Okay, so here you can see there is a diagram which shows what the human insulin production. So you can see that this is the human pancreas. You have isolated the insulin producing gene. Then you're going to use all your recombinant DNA technology along with the vectors and the E. coli, and then you're going to form what, a recombinant bacterium. You just multiply the copies and then insert it into the humans. Okay, so this is the bottle, that is a human insulin which we get. Okay, so this is how gene therapy is being used to cure a particular disease permanently. Then the third one which you have is the molecular diagnosis. So molecular diagnosis is basically of two types. As you can see over here, one is a conventional method, and another one is a recombinant DNA technology along with the PCR and ELISA test. Okay, so conventional method is normally which we all take, that is the serum, blood sample, urine analysis, what we do to check out that if we are having any kind of diseases, right? So it's an early diagnosis of the diseases by using this conventional methods. But in order to go deep, okay, to that particular disease, for example, if it's a cancer, tumor, or something like that, obviously you need to dig up deep, right? You cannot stop with the urine or blood test over there. So you go to what, the next level techniques, which are nothing but the recombinant DNA technology, PCR and ELISA. PCR is nothing but the polymerase chain reaction. It's a process, okay? And ELISA is also enzyme linked immunosorbent assay. It's also what do you say, a particular process which is being used to diagnose a particular disease. Okay, so first let's see about the PCR. So PCR is usually what, polymerase chain reaction. So in order to know or see, now for example, if you have one pathogen infected to that particular human, with one pathogen, if you isolate, you cannot do many tests, right? So you need many samples of the thing. So how are you going to multiply those samples? To do that only, we have this PCR process, that is the polymerase chain reaction, where you can increase the amount of whatever small quantities you're having in your hand. For example, they have given over here is what, a very low concentration of a bacteria or virus can be detected by the amplification of their nucleic acid by PCR. So if you see over here, this is a machine, which is also called as a thermal cycler. In this only, you're going to carry out this polymerase chain reaction. So what do you do? Now this is a in this what you're going to do is you're going to take all the samples which are required to put in this PCR tube, the test tube. In that you're going to take DNA sample, your primers, nucleotides, the polymerase, whatever you require. You put a particular type of buffer of your pH, whether it's an alkaline, acidic, neutral, whatever is there. Then you shift it to the PCR tube and place those PCR tubes into this thermal cycler. When you put it into that when you put it into that thermal cycler, it undergoes the three-step process, which is a denaturation, annealing, and extension. Once you do this, at last you're going to get, if you're just using one sample over there, you're going to get plenty of samples at the end of the process. That is the PCR process in just one cycle. So usually to detect the HIV in the suspected patients, okay, the genetic mutations in the cancer patients, or any genetic disorder is there, or one is there, or more than one is there. So all these things, okay, you can detect by using what? This polymerase chain reaction. Okay, and at last, which you have is the ELISA in the medical thing. So that is the enzyme-linked immunosorbent assay. So it's basically based on the antigen antibody interaction. Okay, so if any foreign particle comes, obviously the antibodies will be formed over there. So this is how it looks like. Okay, this is how the test has been done. You can see over here the preparation, incubation, completion, and finally you will detect if that particular patient is having what any kind of infection. Okay, so that particular antigens can be detected, and you can produce what antibodies against them and insert into that particular patient. Fine.